Welcome to the differential gene expression module. And in this talk, we're going to look at basic tools that are used to study development at a molecular level. We're going to learn about techniques that determine the expression of RNA during developmental processes, as well as techniques that enable the detection of regulatory DNA sequences. Finally, we will learn about techniques that determine the function of genes. There are a variety of tools that are available to enable us to study differentially expressed genes. Now, some of these tools include in situ hybridization that is used to study gene expression, while chip sequencing is used to identify regulatory regions of the DNA that proteins are able to bind to. We also have techniques like deep sequencing that provides us with information on the transcriptome. Additionally, we can look at gene function by performing the ectopic expression of genes using techniques like the GAL4-UAS system. We can also learn about gene function by performing gene knockdown or knockout techniques. There are also microarrays and macroarray techniques that enable us to understand differentially expressed genes during developmental processes.
So let us first look at in situ hybridization. In situ hybridization uses a complementary RNA sequence that's also called as an antisense RNA sequence, and it is used as a probe to a target messenger RNA. The antisense RNA sequence is modified in such a way that the uracil residues of that antisense RNA are conjugated to a molecule called digoxygenin or DIG that enables the visualization of that antisense RNA. In the figure, if we look at the blue boxes, that is where we can see the DIG moiety being covalently attached to the uracil residues of that antisense RNA, which will be complementary to the target messenger RNA that we would like to study. One of the advantages of in situ hybridization is that it is a procedure that can be performed in whole embryos. So to perform in situ hybridization, the embryos are normally fixed with agents like paraformaldehyde or glutaraldehyde and permeabilized to enable the hybridization between the probe and target messenger RNA. Hybridization, as we're going to see in this figure, is when the antisense RNA can form complementary binding with the targeted messenger RNA. After the hybridization occurs, an antibody that can recognize the DIG moiety that is present on the antisense RNA is introduced, and this antibody is tagged with an enzyme, which is usually alkaline phosphatase, and it can bind to the probe or the antisense RNA through the presence of the DIG molecules. Finally, a substrate like NBT or BCIP is added, which is catalyzed by alkaline phosphatase to form a colored product. The color indicates the presence of that target messenger RNA that we are interested in. So when we look at the figure to the right, where we can see this oval-shaped embryo, wherever we see the blue staining is basically where the messenger RNA is actually present. So this is the process by which in situ hybridization occurs. The next technique that we're going to look at is the technique of chromatin immunoprecipitation combined with sequencing, which is also called as chip sequencing. And it is used to identify regulatory regions of DNA that proteins can bind to. Chip sequencing uses antibodies that bind to transcription factors to purify sections of bound DNA, or basically DNA that is bound to these transcription factors. The DNA is then purified and identified by DNA sequencing methods. To perform chromatin immunoprecipitation, The method involves the isolation of chromatin and the cross-linking of proteins and DNA using the agents like glutaraldehyde and formaldehyde. The DNA is then sheared into pieces, followed by the addition of an antibody to the protein of interest. To have a visual representation of this technique, here we can see in the first step where we have our DNA interacting with the proteins in a cell, and the cell will be lysed, and the chromatin that is obtained is going to be cross-linked and sheared. The chromatin is fixed before this whole process occurs to allow the proteins in the DNA to be locked with each other during the shearing process. Once we have the sheared DNA present, an antibody is added to the protein or the transcription factor that we are interested in. Binding of the antibody to the transcription factor is one of the essential steps in this technique. Once that binding occurs, precipitation and purification of these DNA complexes that are bound to the transcription factor occurs. And once we purify those pieces of DNA, the DNA is sequenced. Once the DNA sequences are obtained, they are compared to the genome of the organism to determine where these DNA sequences are present, and thereby this allows us to understand how the transcription factors are able to regulate gene expression. Another method that is used is called as deep sequencing or RNA sequencing. Now, RNA sequencing allows the identification of all transcripts present in a cell, embryo, or in tissues. What it does is it takes advantage of the high throughput capabilities of next generation sequencing technology to sequence and quantify all kinds of RNA molecules that are present in a cell. An example of this technique is shown in the figure to the right, where let's say we want to compare the development of an embryo at 33 hours, 3.5 days, and 6 days. So we obtain cells from different parts of these from the same part of these embryos at the different time points. And then what happens is the RNA is extracted and messenger RNA is obtained, and that RNA is then fragmented to form smaller sequences. This fragmented RNA is then converted to cDNA by the reverse transcriptase enzyme, and then random priming also facilitates the process. Adapters are then ligated and then amplified through the process of PCR, and then sequencing is done. So in this way, we are sequencing all the RNA molecules that have been made in those cells. So we obtain a lot of data in this process. So a lot of sequencing data is obtained. For example, you get tens of millions of sequences per sample. They are then processed to be mapped to different parts of the genome to identify what kind of RNA molecules we are looking at. And ultimately, we are able to define the transcriptome and identify differential gene expression between samples.
In order to determine the function of the proteins that are made by genes, there are different approaches. And one approach is the forward genetics approach, while the other is the reverse genetics approach. In the context of development, a forward genetics approach would include treating the organism with an agent that is known to cause random mutations. And then we would continue to monitor the development of the organism. When we observe a phenotype that is different, then forward genetics requires us to go back to see what genes were mutated, and this way we are able to determine the function of genes. Gene function can also be determined by a reverse genetics approach. And in the reverse genetics approach, we have a gene of interest, and that gene of interest is manipulated by altering its expression in cells, and we determine the effect of those alterations on development. The way the expression of the gene can be altered is by ectopically expressing the genes in different parts of the embryo. This can involve expressing the genes in different places of the embryo and monitoring what happens in those regions. Another way gene expression can be altered is by reducing the expression of the gene. This can be done by knocking out the gene, which implies the elimination of function of the targeted gene. Another way by which we can reduce the expression of the gene is through knockdown strategies that use RNA interference or siRNA to prevent the expression of the gene.
We're now going to look at another system, which is the Gal4-UAS system. The Gal4-UAS system is made up of two components, the Gal4 transcription factor and the upstream activating sequence or UAS that normally acts as an enhancer. So the Gal4 is a protein while the UAS is a DNA sequence. The Gal4 can be combined with the developmentally controlled enhancer as shown in the figure in the blue box. So in this case, the gene that expresses Gal4 is combined with an enhancer sequence, which in this case is the specific imaginal disc enhancer sequence. Thus, in cells that use the specific imaginal disc enhancer sequence, the Gal4 gene will be expressed to form the Gal4 protein, which acts as a transcription factor. Now the UAS, or the upstream activating sequence, is placed upstream of a target gene that can cause Gal4-induced transcription. So if we look at the blue box that is to the right of the figure, here we can see that our target gene is the PAX6 DNA, and upstream to that target gene, we have the UAS sequence. The UAS sequence acts as an enhancer, and it requires the presence of Gal4 in order to cause the transcription of the target gene, which is the PAX6 cDNA. The Gal4-UAS system is used as a powerful tool because it allows researchers to express genes in new places and observe what happens to those new places during development. One way in which we can test gene function is by using the CRISPR-Cas9 system. Now, just to give an introduction, prokaryotes use the CRISPR system to defend themselves against viruses. CRISPR is a stretch of DNA that contains short stretches of viral DNA that, when transcribed, form short guide RNA or shRNA. Now, if you're wondering from where these viral DNA came from, they came from a time when the bacteria were infected with those specific viruses. The short guide RNA that is produced is associated with the endonuclease Cas9 and can also bind to viral DNA that is present in infected prokaryotic cells. The Cas9 can then cleave the viral DNA by generating a double-strand break, and thus it can protect the prokaryotic cell from the virus. We have now been able to use this CRISPR-Cas9 system in all different kinds of organisms, like eukaryotic organisms. So when we look at the CRISPR-Cas9 system in eukaryotic cells, it's mainly used to deactivate genes. The way it's able to do that is we generate or design short guide RNA for the genes of interest and introduce them into the cell along with the protein Cas9 as shown in the figure in the blue box. The Cas9 that is associated with the short guide RNA then causes double-stranded breaks and form in the target genomic DNA. Now, these double-stranded breaks are formed where there is complementary binding between the short guide RNA with the DNA. These double-strand breaks that have been resulted are usually fixed by the host through non-homologous end joining. Now, non-homologous end joining is not perfect, and usually it results in either insertions or deletions that are called as indels. This is what is basically seen in the blue box in the figure, where we can see that there is either an insertion or deletion in the DNA through when we have non-homologous end joining, which is a DNA repair process. The creation of these indels causes a frame shift or a nonsense mutation, and thus the gene can no longer be expressed because the protein that is produced is simply not functional. CRISPR-Cas9 has also been used for homologous DNA recombination. However, we won't be looking at that process in these slides.
The CRE-LOX system is used for knocking out genes in an inducible manner, and thus we are able to control when the genes will be knocked out in the organism. There are two components to the CRE-LOX system. We have the enzyme called as CRE recombinase and DNA sequence called as LOXP sequences. The CRE recombinase is able to remove any sequences that are between the LOXP sites. Now in the figure, we can see the CRE recombinase is under the control for an enhancer. Now in this specific example, it is under the control of an enhancer for a protein called as albumin. When we have transcription factors that regulate the expression of the albumin gene, in that scenario, CRE recombinase will be activated. We're looking at the deletion of an allele of HNF4 alpha, which is a gene. So in the lower figure, we're looking at the exon sequences for HNF4 alpha, and 2 LOXP sites have been added on either side of exon 2. These LOXP are binding sites for the CRE recombinase, and so we are trying to delete the HNF4 alpha exon 2 DNA sequence in this example. Now in most cells, albumin is not expressed because the transcription factors that would bind to that enhancer are not present in the cell. However, in the case of liver cells, albumin is expressed because the transcription factors that are able to bind to that enhancer are present in liver cells. Thus, in liver cells, we will be able to see the expression of CRE recombinase because it is under the control of the enhancer for transcription factors that will regulate albumin expression. When the CRE recombinase is expressed, it is able to bind to the LOXP sites, and it forms this looped structure as shown. Now, depending on the orientation of the LOXP sites, we can have a deletion, which is what we are seeing in this example. In other cases, the orientation of the LOXP sites can also facilitate the insertion of DNA sequences. Thus, the CRE-LOX system can cause deletions or tissue-specific knockouts or even misexpression of genes. RNA interference is a very popularly used technique in order to knock down gene expression. Now, gene expression can be inhibited by using small interfering RNA or siRNA or short hairpin RNA, which are also called as shRNA. By using siRNA and shRNA, we can inhibit gene expression by preventing translation of the messenger RNA that is formed or promoting the degradation of the messenger RNA. Now, silencer RNA are small double-stranded RNA, and they can be synthesized commercially and introduced into cells in order to knock down the gene of interest. On the other hand, short hairpin RNA or shRNA are introduced into cells as DNA sequences in a plasmid that will then be transcribed into the short hairpin RNA. These short hairpin RNA that are synthesized by the cell undergo cleavage by an enzyme called as dicer, and they end up forming the small interfering RNA. Thus, short hairpin RNA will ultimately form small interfering RNA. This double-stranded RNA sequences that are short tend to be separated by an enzymatic complex called as RISC. This enzymatic complex will separate the double-stranded RNA and will associate with one of the strands. Now, this strand of RNA can then bind to target messenger RNA that have complementary sequences. This binding of the RISC complex that has that single-stranded RNA to the target messenger RNA can result in either the ribosomes being stalled, due to which translation does not occur, or we can have the degradation of the messenger RNA. And either way, we will not see the expression of protein. Thus, even though the gene is present in the cell, the gene is not expressed due to the effects of the small interfering RNA or short hairpin RNA. With this, we come to the end of our talk, where we learned about techniques that allow us to determine the expression of RNA during development. We also learned about techniques that enable us to detect regulatory DNA sequences that transcription factors can bind to. And we also learned how to determine the function of genes using a variety of techniques.